And good afternoon, everyone. My name is Doory, Assistant Professor in the Department of Chemistry at Hanyang University. So first of all, I want to say that I really appreciated this opportunity to present my recent work in this wonderful workshop. So I joined Hanyang University 3 years ago, and the title of my lab is Super Resolution Spectral Microscopy Lab. So my research specialty is super resolution microscopy, and this technique overcomes the resolution limit of the traditional light microscopy. So with this technique, we could resolve the many nanostructure inside the cell, which cannot be resolved by the conventional light microscopy as shown here. So today I will present our recent work on the application of this technique to the small size platelet. In particular, I will present the super resolution imaging study on the molecular mechanism of platelet activation process and release process respectively. So before we talk about our main story, which is a platelet, let me briefly introduce our recent work, which is about the development of a new approach for the super resolution fluorescence imaging with ultra-low laser power. As I said, the STORM is based on the fast photo switching property, and to obtain this, high laser power is required. However, as you know, high laser power would be very harmful for the very weak biological sample. So to overcome this limitation, our lab was trying to develop a new approach for the STORM imaging with ultra-low laser power. So before we develop this method, we tried to systematically characterize the various photo switching properties on the excitation laser power. And then we found that as the laser power decreases, the photon number decreases and the resolution gets worse and the photo switching rate gets lower. So because of that, we could see a lot of overlapped molecularization at a low laser power, which makes the low image quality, low image quality like this. So to overcome this limitation, we try to find the best imaging buffer condition for the ultra-low laser power. So from the test of the various SIO reagents, SIO concentration, pH refractive index, and anti-fading agent, we could find the best optimized imaging buffer condition for the ultra-low laser power. And we found that under this optimized condition, we could increase the photon number more than twice, and we could increase the photo switching rate more than 3 times. And then under this condition, under this optimized condition, we could increase the STORM image quality significantly as shown here. This work was recently selected as the cover article in Analytical Chemistry. Then let's talk about our main story for today, which is the platelet. As you know, platelet is one of the most abundant components in blood, and it is responsible for blood clotting. When bleeding, platelet becomes activated, but the ultrastructural changes of platelet during this process has not been known. So we try to understand the ultrastructural changes of platelet during this activation process. So we conducted this ultrastructural study for the 2 important platelet process. One is platelet activation, another is platelet division. So let's talk about them one by one. First of all, platelet activation. So first of all, we try to observe the activated platelet in our experimental condition by using PMA as the agonist. So first of all, we tried the low resolution of the live cell DIC image for the activated platelet by PMA. And from the live-cell DIC image, as you can see here, we could observe that the round shape of the platelet started to be spread out upon activation like this. But as you can see here, the resolution is very low. So to observe the ultrastructure changes of this process, we perform the live-cell three-dimensional optical diffraction tomography using a tomocube device. And from this experiment, we could observe a very interesting two phenomena like this. First of all, dynamic protrusion like this. Another interesting phenomenon was ballooning effect like this. These two interesting phenomena are known to be the representative activation process of the platelet. So from this observation, we could confirm that in our experimental condition, the platelet are activated very well. So under this condition, we try to take the storm image to observe the ultrastructure of the various organelle inside the platelet. So first of all, we took storm imaging for the cytoskeletal elements, among them, first of all, actin. So when we took storm imaging for actin filament, we could observe that in resting platelet, we could observe the mesh network, homogeneously distributed mesh actin network. And as the activation process is going on, this platelet is stretched out in one direction, and then finally radially spread out as shown here. This ultrastructure has not been resolved by the conventional light microscopy. And from this storm imaging, we could also quantify the directionality of the spreading, as shown here. We also quantify the actin nodule because we could see a lot of actin nodules as the activation process is going on. And we found that the number and the size of the actin nodule increases during this activation process. We also found that when we measure the centroid of this actin nodule, it is consistent with the center of mass of the spread platelet in activated process. So we found that the activated platelet can be spread out with the help of the actin nodule. So in conclusion, actin filaments seem to control the overall shape of the platelet. Next, we took storm imaging for microtubules. As you can see here, as the activation process is going on, we could observe that the ring shape of the microtubule ring started to unwind and then form the small ring structure on the center at the end of the activation process. This ring shape of the microtubule is called the marginal band. And it was also, the similar pattern was also observed from the correlative storm and SEM imaging, as shown here, which reveal the relative position of the microtubule with respect to the overall morphology of the platelet. Based on this, we try to investigate the role of the actin and microtubule more in detail. So first of all, we studied the role of the actin filament by depolymerizing actin using cytocollagen D. And as you can see here, from resting sample, there's no big difference between the control and actin depolymerized platelet. But from the activated actin depolymerized sample, we could observe very interesting this square shape of platelet. And from this image, we could also observe the microtubule bundles at the four corners. So, but it is EM section 2D image. So to see the full 3D structure for this, we took 3D storm imaging like this. And from the 3D storm image, we could see this interesting potato chip-like structure. So based on this, we could confirm that actin depolymerized platelet could not be spread out on the covered glass. and a microtubule event with flexibility. So in conclusion, actin, it seems that actin is responsible for providing the spreading force, and then microtubules tend to be flexible during this activation process. Then next, we studied the role of the microtubule by depolymerizing the microtubule using nokodazole drug. And as you can see here from the resting sample, we could see this spiky shape of the platelet from the microtubule depolymerized platelet, probably because of the absence of the microtubule marginal band, what I showed you in the previous slide. And from the activated sample, we could observe a little bit shrinked platelet from the microtubule depolymerized sample with a smaller number of the actin nodule like this. So we could conclude that the microtubule can control the actin nodule during this activation process with increased flexibility. So next, we try to take the storm imaging for another cytoskeletal element, which is spectrin and Vimentin. And as you can see here, we could observe that homogeneously distributed spectrin are concentrated in the center of the activated platelet at a relatively high position from this XZ cross-section of the 3D storm image. In a similar way, Vimentin also show this centralization in the kind of yolk region of this fried egg morphology with increased density during this activation process. It implies that this membrane skeleton probably maintains this convex shape in the center to regulate the centralization of some organelles. So from this observation, our next question was then, what kind of organelle would be centralized under this central region? So to check this, to find them, we took storm imaging for various organelles. We took first of all, we took storm imaging for the mitochondria. And interestingly, we could observe that these homogeneously distributed mitochondria are transported to the middle central part of the activated platelet with an increased their size and number. Interestingly, DTS, in other words, the ER in platelet also shows a similar pattern. So upon activation, DTS shows the centralization. And in a similar way, autophagism also shows a centralization upon activation as shown here. So in conclusion, all of the double membrane organelle, including mitochondria, ER, and autophagism shows this centralization upon activation. We also were wondering the distribution for the secretory granules, because secretory granule is very important in platelet. They have hundreds of proteins inside them, and they trigger exocytosis of their contents during activation for the bleeding signal. So we took imaging for the alpha granule and dense granule. And interestingly, again, we could observe the centralization of the alpha granule upon activation. And in a similar way, dense granule also shows the centralization as shown here. So in conclusion, all of the double membrane organelle and secretory granule show the centralization upon activation. Then our next question was what triggers decentralization of organelles? So one possibility could be the formation of this small sized microtubule ring, what we observed from the STORM image. So to test this, we try to depolymerize this microtubule. If our assumption is right, we may not be able to see decentralization from the depolymerized sample, microtubule depolymerized sample. So to test this, we took STORM imaging for the various organelle after depolymerization of microtubule. But unexpectedly, as you can see here, we could not observe any significant differences in their centralized location from this microtubule depolymerized sample. It suggests that the formation of the small ring of the microtubule at the end of the activation process is not likely to be the main force of the centralization of organelles in this activation process. So based on this, we thought there might be another reason why they are centralized. So one possibility, another possibility could be that there could be the existence of the exit route on the center for the protein, a secreted protein from the organelles for the bleeding signal. So for example, OCS vacuole can be the good exit route for the secreted proteins. So we try to observe where the OCS vacuole can be formed at the end of the activation process. So to observe them, we took STORM imaging for the OCS vacuole by using the membrane staining. And fortunately, by using the membrane staining, the non-red staining, we could observe the OCS vacuole in super resolution for the first time, as shown here. And from this STORM imaging, we found that the number and the size of OCS increases during this activation. We also found that the OCS in the activated platelet is mainly localized in the center of the platelet. This location of the OCS vacuoles can be also observed from the EM section image like this. As you can see here, as the activation process is going on, the number and the size of the OCS increases. And at the later stages of the activation, most of the area in the yolk region of the fried egg-shaped spread platelet were occupied in OCS, and they were surrounded by the various organelles such as mitochondria, ER, autophagosome, and granules. So next, to observe all of them together, we took a high voltage EM tomography of the central region of the activated platelet. In other words, the yolk region of the spread platelet. And as you can see here, we could observe that the yellow and the blue color alpha granule and dense granules are closely surrounding the OCS, enlarged OCS, and many of them are in contact with the OCS, which probably implies the formation of the route of cargo release. So as you can see here, the close association of the granule and the OCS could be observed from the high voltage EM image. Like this, granule membrane fusion to OCS will trigger the granule secretion. So in summary of this part, we found that from the STORM image, we found that actin can provide a main force for the spreading, and it can determine the overall shape of the platelet. And we also found that microtubule can play a role in the formation and rearrangement of the actin nodule. in this process. And spectrin and Vimentin can support and stabilize the extended structure of the central bump region, in other words, the yolk region of the fried egg. And under this central bump region, we could observe that double membrane bound organelle and granules are centralized. It is because of the granule membrane fusion to the OCS vacuole to trigger granule secretion. Then let's move on to our second topic, which is platelet division. As you might know, although it is known that this platelet does not have nucleus, it was recently suggested that it can be divided into platelet division, platelet progeny, to control their numbers through these various platelet intermediate. So we try to understand the ultrastructure changes of platelet during this process by observing various platelet intermediates. So this work was selected as the cover article and structure this year. So first of all, we try to observe whether we try to test whether we can observe the various known platelet intermediate in our experimental condition. So to do that, we use again the membrane staining, and fortunately, from the membrane stain platelet sample, we could observe various known platelet intermediate. For example, resting, circular preplatelet, oval-shaped preplatelet, barrel-shaped proplatelet, or multibody proplatelet. And they were also observed from the live cell holography, as shown here. And from this live cell imaging movie, we could also quantify their relative population. And from this quantification, we found that as the culture period is increased, the relative population for these intermediate decreases. But the relative population for the resting platelet increases, which implies the platelet in our experimental condition can divide. In other words, the platelet release in culture. So based on this, our experimental condition, we also try to observe the interconversion between the different platelet intermediates. So to observe them, we use the live cell holography. And from the movie, we could observe the interesting interconversion. For example, the conversion from the proplatelet to preplatelet, like this, by observation of one body to another, like this. And there's another movie for the conversion for from the proplatelet to preplatelet, like this. We also use the live cell fluorescence imaging for microtubule. And from this fluorescence movie, we could observe the conversion from the proplatelet to preplatelet by folding of the microtubule band. And in another movie, we could also observe the conversion from the preplatelet to proplatelet, like this, by twisting of microtubule band. Like this, we could observe various kinds of interconversion between the different platelet intermediates. So there, it seems that there are many different pathways for them. So based on this, we try to observe the ultrastructure changes of the organelle during this division process. So first of all, we took storm imaging for the microtubule. So first of all, in resting platelet, we could again observe the ring shape of the microtubule, in other words, the marginal band. And then as the division process is going on, it started to be unwound and elongated. And then finally form the multibody like this. They are connected by the bridge, and in a bridge, we could observe the twisted microtubule, and it was not resolved by the conventional light microscopy. So from this, we could confirm that microtubule can control the overall shape of the platelet intermediate. And next, we also took the storm imaging for other cytoskeletal element, actin and spectrin. And as you can see here, both of them show consistently dense and homogeneous distribution, regardless of the intermediate. And in contrast, the double membrane organelle showed a different pattern. For example, DTS were separately distributed on the other side of the barbell-shaped proplatelet, as shown here, by associating the membrane. In a similar way, alpha granule also show the equal division in the bubble-shaped proplatelet and multibody proplatelet to prepare their equal division. And dense granule also show this confinement to the platelet-sized swelling in bubble-shaped and multibody proplatelet. So it seems that they are also preparing for their equal division. So we again took the high voltage EM tomography to observe all of them together. And as you can see here in this oval shaped pre-platelet, we could observe that DTS goes to the membrane, plasma membrane first, to prepare their division, and then granule go to the membrane, and then mitochondria go to the edge before their division. So from this observation, our next question was then what kind of who help this transportation for the division? So one possible helper could be the microtubule or actin, because they are well-known mediator for the protein transportation. So to test this, we depolymerize the actin or microtubule by using the cytocollagen D and nocodazole. And from this test, we could observe that when we depolymerize the actin, by using cytocollagen D, alpha granule and DTS showed the biased distribution. It suggests that actin can play an important role in guiding the homogeneous distribution of alpha granule and DTS during this division process. In contrast, when we depolymerize the microtubule by using the nocodazole, mitochondria and dense granule show the biased distribution as shown here. So in conclusion, microtubule might derive the homogeneous distribution of the dense granule and mitochondria during this process. We could observe the similar biased distribution for in the barbell-shaped proplatelet from the EM section image as shown here. So we could conclude that the distribution of DTS and alpha granule can be governed by the actin, whereas the distribution of the mitochondria and dense granule can be governed by the microtubule. So in summary of this part by using the live cell phase microscopy, we could observe the interconversion dynamics between various platelet intermediates. So it seems that there are multiple different pathways for the interconversion between them. And from the storm imaging of various organelle inside this platelet, we could confirm that microtubules are responsible for controlling the overall shape of the platelet intermediate. And from the study of the effect of the cytoskeletal element on the distribution of organelle, we found that the distribution of DTS and alpha granule are governed by the actin, and the mitochondria and dense granule are governed by microtubules. So this is all for today's presentation. Thank you very much for your attention.